Good evening. I'm going to do a video that's a little bit different from some of the other videos that we've done before, because this video is a draft, literally. I am going to think about writing up some of these findings. And so I want to talk it through, so I figure I might as well try to talk it through to you as a video. And also this will be a good way for me to communicate to my students who put in a lot of hard work on this, and I want to show them what their hard work resulted in. Basically, we did some research on the computer that it supports the hypothesis that there's considerable cross-reactivity in the T cell responses to coronavirus. Let me explain a little more about what that means, but there are other videos where I go into a little bit more detail. I'm going to break this into two parts. The first part I'm going to be talking more about background and methods, and then I'm going to be talking about connecting it to the experimental results after that. So that way you can definitely tune out at any point, really, but you can tune out before part two if you're not that interested in how it lines up with the experiment. So I have two levels of a title for this. Cross-reactive immunogenicity of coronavirus T-cell epitopes. And even though that sounds like a mouthful, it means what it says. There's cross-reactive nature of the immune system going on here. It's happening in the T-cell level, and it's happening against coronavirus. So I can translate that title a little bit to say that T-cells can recognize the shapes that are in common between SARS-CoV-2, the coronavirus virus, and common cold coronaviruses. And this is a theme that I've talked about a little bit before. Now I'm having it all in one place, so I can talk about the research project. So previously on the data updates, you can go back and you can look at these if you have questions, but there's a big mystery, a good mystery, that's been coming out of the scientific literature. There are places where they test T cells for people who have never been infected with SARS-CoV-2. They've never had a chance to see the virus in some cases. And yet 30 to 80% of the T cells, depending on how you measure it, honestly about half is what I'd say, about half of the people have T cells that will react weakly to little bits of SARS-CoV-2. This is interesting because the T cells are holding a memory of past infections. And somehow, you've never seen SARS-CoV-2, but you might have T cells that respond to it, that remember it. How can they remember something they've never seen? Well, there's actually a scientific reason for all of this. The reason is there's no virus. No virus is an island, okay? Viruses are all related to other viruses, and you've been infected with their family members before. So you might not have been infected with this exact virus, but you might have been infected by its cousin. For that reason, a T cell can actually recognize something that is not exactly the same as what it's seen before. T cells are deliberately messy. They can recognize something, they only recognize maybe, 50 to 75% of the shape that they're looking at. And they don't care about the other, 1/3 to 1/2 of it. There's actually a reason for this, because if the virus mutates part of that shape, it won't automatically evade the immune system. It might be part of the shape that the T cell doesn't care about. So this is actually a good thing. Your immune system is messy and doesn't care about every little detail in the virus. It just cares if it recognizes the general shape. Because coronaviruses share some general shapes, there is a possibility that you'll have a T cell that recognizes a general shape from the common cold, and that same general shape is on the SARS-CoV-2 virus. That's how it works, and the metaphor is, if you can recognize cousins that are 12.5% genetically identical, a T cell can recognize virus cousins that are 50% genetically identical. There's a lot more to that than that metaphor, but I'll talk about some of that. Some of this has been discussed in previous data updates and previous videos I've done, so I send you to those if you have questions about the background. The main point, if I can summarize those in one slide, is that the common cold coronaviruses are actually related to this SARS-CoV-2 that we have. If it is shown here boxed in yellow, the SARS-CoV-2, and this is in its family tree. So you have cousin viruses, and if you look down at that family tree at the bottom, you can see that there's bat viruses are like brothers and sister viruses to the SARS-CoV-2. But then a little bit farther out on the family tree, you see the ones in red boxes. All of those are HCOVs. That means human. And these are circulating. They're not all of the common colds, but they're a significant fraction of the common colds are one of these 4 coronaviruses. So this would be the hypothesis. Can we find parts of the genomes of these common cold viruses that look like the SARS-CoV-2 genome? Stands to reason that they exist, but the question is how much, how many, and how closely do they resemble in the way that we look at it as a virus looks at it. If you look up here, you can see that there's about 40 to 50 to 60% identity. That means the genomes are about 40 to 60% identical among all this family tree. And they're even more identical when you compare, like, for example, the HKU1 cold virus to SARS-CoV-2. And so when a virus invades your cell, it hijacks the whole machinery and it makes more viruses. It takes over the copy machine of the cell, and it makes a bunch more viruses, and those take over your body. The cell of the body uses this against the virus because it'll actually have a system for a molecule that shows what's on the inside of the cell. It's called the MHC molecule, and this was the subject of my graduate research. Here's an MHC molecule right here bound to what's called an epitope. An epitope is a short little snippet of a protein. It's also called a peptide. And it's just a really short little bit, like a curl of its hair, almost, that it's holding up. It's bound to this MHC protein, and it's put on the surface of the cell. And T cells come along, and they look at these. They look through the MHC molecules to be able to try to find if there is an epitope, a peptide, that they have not seen before. If there is, then they react against it. If there is, then they remember that shape so they can react more quickly against it next time. So this helps T cells respond, and the fact that they're messy, notice how this epitope only presents one side of it. If you're a T cell looking down at this red peptide, you can only see the front part of it. The bottom part of it, the back part of it, you can't see at all. It doesn't matter if the shape of the back part changes a little bit. That's what I'm saying when I say that viruses don't have to be identical for the T cells to recognize them. They just have to present the same surface. They have to look the same from this perspective. And in general, we say that means they have to be about 50% to 67% identical when we look at the protein sequences. And that's all the biochemistry that we can get into some other time. The reason why this is important is I actually heard about this way back when, before any coronavirus was even 2019 was even a glimmer in somebody's eye, okay? This is back in like 2012, 2013 that these studies came out. And this is a study of human immunology. Human immunology likes to work like this. It likes to be a little bit messy. Because if you develop immunity against one virus, you can apply that same immunity to another virus that may be very different shapes in other places. But if it has the same shape in that one little place that you're looking at, then you can activate against it. In this way, you can have immune cells that react to one kind of virus, and then they turn around and look at another virus with a similar shape. It could be a very different virus. As long as it has a similar shape in that one little bit, it can react against it. Here's the evidence for some cases of this. These are 5 different T cell clones, that's actually the name of them, that react to the HIV peptides. If you take these T cells and you show them these different little peptide snippets, right here, these are the exact peptide names, and look at the names of the diseases that these come from. A. tropicalis, P. marinus, L. bacterium, B. bifidum. None of these are things that the T cells would have been able to see before. And yet the T cells are reacting. You see all those pluses on the right side of things. That means that the shapes of these short little sequences are similar enough to HIV shapes that HIV T cells are able to recognize them. Basically, this is evidence that there are 5 different TCR clones that will recognize new peptides from weird bacteria if they were educated on HIV peptides. This is a deliberate messiness that works in your advantage because you've never seen A. tropicalis, but now you have at least one T cell that reacts to it. This is called cross-reactivity or cross-immunity, and it's what we're talking about. Here's another case. So flu vaccination activates, it produces T cells that remember what flu looks like. Well, they found particular cases where they have F. magna and T. vaginalis here. And those have sequences. If you look at those letters down there, those letters are almost the same as the flu letters on top. So it stands to reason that if you have T cells that respond to the flu letters, they'll also respond to the F. magna letters. And that's what they found. They found because the T cells recognize these short little snippets represented by the letters, they actually can recognize things that they've never seen before if the snippet is the same. And if the genome is similar, you're going to have more snippets that are the same. It actually works exactly the same way as if you're cutting and pasting text. And the good news is this text, the text of RNA in the coronavirus genomes, only has 4 letters. So there's a good chance of some cross-reactivity because there's a good chance that you'll have similar letters in similar places in different viruses. Okay, so that's the theory. And I've shown this data before. This is from the Grafoni et al. Paper that sort of started me off down this path. And I want to point out that here it shows, on the top, it shows 2 different kinds of T cells. There are 2 branches of T cell immunity. CD8 positive class 1 immunity, CD4 positive class 2 immunity. And if you look at those, if you look at SARS-2 infected T cells from those patients, you'll see that there's a lot of high points on that. There's a lot of T cells from different patients. They have some T cell somewhere that's reacting to these little bits of protein. So, and you can see that those dots are high. This is one of the first evidence that we had that the T cell response to SARS-CoV-2 is actually very strong. At least strong. I don't know enough to say very yet, okay? But if you look down at the bottom, so on the top you have the SARS-2 infected, and you see all those T cells reacting right there. Here's class 2 T cells reacting. They're reacting even more. Across the board, you see a lot of the points that go up. Down below, they took blood samples from people from 2018, before SARS-CoV-2 ever made the jump from animals to humans. And look at that. You see fewer dots that are high, but you still see several dots that are high. There are some T cells that respond, and this is in the class 1 branch. If you look over to the right, the class 2 branch also has T cells that respond. This is where they got when they broke this down by different people, they said at least 20% of people have class 1 T cells that will respond. At least 50% of people have class 2 T cells that will respond. It comes out to be their numbers about 60% of people will have one kind of these or another. And other people have come to an estimate around 50%. I had some of those papers in my July 4th edition of this. So what did my students do? We're working with the letters, and we have all the letters in the SARS-CoV-2 genome, then we have all the letters in the 4 coronavirus common cold genomes. And what we do is we have a program that sticks them next to each other, and we look up and down on that program, look up and down on that stack, and we say, does it look like the same letters in the same place? If it has the same letters, but above a certain threshold, which I'll talk about in a second, then we'll say that it's similar enough that the T cells can recognize, if they're educated on the common cold, they can possibly recognize the SARS-CoV-2 epitope. So you can actually, this is actually the workflow that they did, and I'm going to put this online so that people can participate in it. If you're interested in finding the information for when I put it online, do shoot me an email, let me know. So basically what you do is we have the computer predictions of all the little epitopes that are going to be seen by the immune system from SARS-CoV-2. So you look up the SARS-CoV-2 letters, and you find that in the SARS-CoV-2 lineup that we have right here. Then what you do is you look up and you see, oh, okay, There's the OC43 sequence right there. It's pretty similar. There's a lot of letters that are the same there, but is it similar enough? So this one actually is chemically similar, but this is not quite similar enough for our threshold. Our threshold is fairly stringent. And so they copy and paste the letters into an Excel sheet. We actually use Excel. It's kind of embarrassing, but we do. And they note where the residues start and stop. You look up the numbers, and you write them down in the sheet. You count how many residues are identical, and you say, from that, we'll say that this one is, I'm not sure how percentage this is probably about 40% identical. Pretty close, but not close enough for a T cell to recognize. When you get 60 to 70% of the letters the same, the T cell can very possibly recognize that same thing. And so we go off that percent identity. So this is all done by students, and I want to give them a huge shout out and thank you. And several of the students, in fact most of the class, went on after the class to do the bulk of the work over the summer. So this is actually a good coronavirus quarantine research project that we were able to do. Here's some examples of a case where we have potential cross-reactivity among 3 viruses, SARS-2 and the 2 cold viruses compared to it. Below you have a case in the black box where there's no predicted cross-reactivity. They're kind of similar, but they're not similar enough to trigger our threshold. And all it is counting letters. So it was kind of tedious. Research isn't necessarily exciting adventure, but when you get to the end of it, you actually have some really interesting things that you found, just like with us. By the way, how do we tell when it's similar enough for T cells to recognize? Well, we have experimental data in each case for a similar situation where we set the thresholds based on the experiment. Here are the 2 papers that we got our thresholds from. These are the thresholds we got from them. I can talk into more detail if you're interested about exactly where in the papers these come from. But these are supported by experimental data. We set the thresholds, and then we see that they go with these experiments and with other experiments. The results that we get out of it go with the experiment. That's what the second part is going to be about. But before we get to that second part, I want to tell you how much of the common cold genome is similar to the SARS-CoV-2 genome. Well, here's the class 1 similarities. The white circle is all of the little snippets, all of the epitopes that can be recognized by the immune system. 793 little snippets. Out of those 793, you have this many viruses, this many for each of the 4 common cold viruses. Each of them is a different color. 283 of the epitopes have match at least one of the common cold sequences above our threshold. That means 35% or more than 1/3 of the SARS-CoV-2 sequences, epitopes, match a common cold epitope. And you can see how much overlap there is between the different viruses. 52 of them are found in all 4 common cold viruses. So again, you don't get every one of these viruses, and not every cold is a coronavirus. But those that are, you have about 1/3 class 1 similarity. Did the same thing for class 2. There's more peptides that we find for class 2. There's 1,045 peptides that we found in the computer. Out of those 1,045, about 1/4, 248 out of the 145, match at least one common cold. And you see in the middle there, 65 of them match all 4. And so that means that about 1/4 of the epitopes for class 2 can be recognized from both SARS-CoV-2 and common cold. So when you compare the 2 circles, you actually see some interesting differences. When in absolute terms, they actually are about the same, but remember that there are more class 2 epitopes. So that means that the class 2 coverage is smaller. It's about 1/4 than the class 1 coverage if you have all 4 of the common colds. But because class 2 has more overlap among the strains, if you look over here, there's more, those circles are closer together on the left. So class 2 is actually going to have more overlap, meaning if you get just one of these 4, there's still a pretty high chance, there's actually a pretty fairly similar chance that you will get one of these like 65 shared epitopes. And so the overlap between them is more common. So what this all means, if you notice all of these, they come down to each time you catch a coronavirus common cold, you get the chance to see about 1/10 of the epitopes that will allow you to react to SARS-CoV-2. How many of those are important? We have to look to experiment to find those. That's the next part. But each single common cold strain, each single different one you get infected with, gives you about 10% of the information that you need to completely see all of SARS-CoV-2. And you don't need to see all of SARS-CoV-2 to fight off SARS-CoV-2. This is actually pretty good cross-reactivity. It's not as high as flu cross-reactivity, but it's about what you should expect based on the sequence similarity. It's about like recognizing cousins. And if you get more than one common cold, then you get another 1/10, and those 1/10ths overlap a little bit. So basically, if sometime in your life you've caught all 4 common cold strains, you have a chance of seeing 1/3 of the class 1 epitopes and 1/4 of the class 2 epitopes. And this seems like it's the kind of thing that could give you some good T-cell immunity. Note that this is different from immunity from infection, and immunity is a sliding scale, not an absolute, like courtroom immunity means a different thing than T-cell immunity. So flu strains have more cross-reactivity because their sequences are similar. Their genetic sequences are like 90% identical in many cases, whereas coronaviruses are more around 60%. But still, on the other hand, actually, coronaviruses don't mutate that quickly relative to other viruses. They're kind of big and slow mutating, which is, again, good news. You can see about that. So the cross-reactivity sort of has a double-edged sword, and you've got to take the whole virus the way that you're given it. Now, one thing I should remember to point out again, cross-recognition is not always good. There are some cases, there are things like antibody ADE, which is an antibody-based crossed immunity, where you can actually have a bad effect from recognizing a virus again, from recognizing cousin viruses. That's the bad news. The good news, on the other hand, is there's only one case of coronavirus cross-reactivity I've found, which is a weird cat coronavirus. Doesn't seem like ADE is too common for coronaviruses. It's more common for something like dengue fever. So how do these compare to experimental epitopes? Better than I thought they would, honestly, because we're doing all of our experiments on only one type of HLA-MHC molecule. All of these experimental data, except for one experiment, but all the ones that come from natural populations, have a mix of different HLA-MHC haplotypes. We only looked at one, so we know that there's stuff that we're missing, that people will be able to see. We're approximating a complex population with the most common member of the population, but still it's not going to capture everything. Even with that, look at these numbers. Cross-reactive epitopes were found in 15 of 17 class 1 peptide pools, in 148 of 248 class 1 epitopes, including 26 of the 28 structural protein epitopes. Those are high numbers, especially compared to, remember that the cross-reactivity is about 30% of the possible epitopes, but it looks like that 30% is more immunogenic. It's leading to immune responses that we can measure in the lab. I think that there's a cause and effect relationship here. I think it's possible that the T cells are being primed by previous coronavirus infections. But that is hand-waving right now. For right now, I simply must show you the correlation and leave discussions of causation up to your own imagination for now. When we get to class 2, we have a little less of the percentage, but it's still substantial, still more than half. 11 of 17 peptide pools, or 5 of 9 cross-reactive peptides. And then we have 4 of 5 class 1 epitopes, which is the Chower et al. That I mentioned before. I mentioned that experiment. That's the one experiment where we have a defined single HLA-MHC class 1 molecule. And in that case, we have an 80% overlap between cross-reactivity and natural observed experimental reactivity, including reactivity in the healthy donor who is, as far as they knew, never been infected with SARS-CoV-2. Okay, so again, I'm trying to explain this, so I'm going to talk about each of these experiments in the next part. If you've stuck around this far, thanks for listening. I hope that I've shown you that cross-reactivity is substantial. We don't quite know yet what this means. Like I said, there's a small chance that this could be bad. But this is more cross-reactivity than I expected when we started this, and more experimental confirmation of that cross-reactivity than I expected, suggesting that the cross-reactivity is experimentally important to real-life immunity. So you can see why I want to talk this through. Thanks for listening, and stick around for part 2 if you want to do that.